Okay, so this is concept 7.4. During oxidative phosphorylation, chemiosmosis couples electron transport to ATP synthesis. This cellular respiration pathway produces the most ATP. So the molecules of NADH and FADH2, which are electron carriers and were used during glycolysis and the citric acid cycle, donate electrons to the electron transport chain. The electron transport chain powers ATP synthesis via oxidative phosphorylation. The electron transport chain is the first part of oxidative phosphorylation. It is a collection of molecules in the inner membrane of the mitochondria. Most of the components of the chain are proteins, which are bundled together into multiprotein complexes. Electrons are passed through this chain sort of like a staircase. Each electron carrier alternates between reduced and oxidized states as they accept and donate electrons down the chain. And these electrons drop in free energy as they go down until they are finally passed on to oxygen, which then forms water. Some of the proteins that make up the chain are called cytochromes, which are proteins with an iron atom. And the electron transport chain generates no ATP directly. Rather, it breaks large free energy from fuel to oxygen into smaller steps, so they can release energy in smaller and more manageable amounts. So this is just an overview of what I said earlier. This is the last cellular respiration pathway, or like the last major one. In the chapter, and it takes electrons from glycolysis and the citric acid cycle in order to eventually produce ATP. This is figure 7.12, and it shows the electron transport chain. You can see groups of protein bundled together into complexes. And the final step is for the electrons to get transferred to oxygen, which then produces water. And throughout this process, free energy drops. This is the second part of oxidative phosphorylation. The actions happening in the electron transport chain causes proteins to pump hydrogen ions from the mitochondrial matrix to the inner membrane space. These ions then move across the membrane and pass through the ATP synthase, which is a complex of membrane proteins that synthesizes ATP. ATP synthase uses chemiosmosis, which is using energy of the hydrogen ion gradient to drive cellular work in order to drive the phosphorylation of ATP. So this is figure 7.13. It's two images of ATP synthase, which is in the inner mitochondrial membrane. But basically it works like a watermill, sort of. So first, hydrogen ions flow down their gradient and enter the stator, which is basically a channel hooked up to the rotor. And there are binding sites within the rotor. And once the ions are binded, this changes the shape of each subunit inside the rotor, and it makes the rotor spin. Each hydrogen ion makes one complete turn before leaving the rotor and entering the mitochondrial matrix. The spinning of the rotor causes an internal rod hooked up to it to spin, which then activates the catalytic knob it's also hooked up to. That actually creates ATP, and it bonds ADP and an inorganic phosphate together to do that. And this is a computer model of ATP synthase. So chemiosmosis is an energy coupling mechanism that uses energy stored in the form of a hydrogen ion gradient across a membrane to drive cellular work. The hydrogen ion gradient is referred to as a proton motive force in this process because it has potential energy and it's able to perform work. The energy used couples the reactions of the electron transport chain to ATP synthesis. So this is figure 7.14. It's basically an image of oxidative phosphorylation. As you can see, it's split into sort of two steps. So first, there is the electron transport chain, which is obviously transporting electrons until they eventually make water. And through this process, hydrogen ions are pushed out across the membrane, and through their gradient and chemiosmosis, they are put into ATP synthase and eventually create ATP. So during cellular respiration, most energy flows in this sequence. The energy from glucose transfers to NADH to the electron transport chain and then to the proton motive force, which ultimately creates ATP. About 34% of the energy in a glucose molecule is transferred to ATP during cellular respiration, making about 32 ATP. The rest of the energy is lost as heat. There are several reasons why the number of ATP molecules is not known exactly, and it's generally, in essence, because the creation of ATP can differ in every instance. So this is figure 7.15. It's basically an overview of everything we've already talked about. And here's oxidative phosphorylation in all its glory. It creates the most ATP out of the three pathways. But all of these pathways ultimately depend on each other to make all of them work.